Hello everyone, my name is Harnish Shah and this is my final project for cell biology class. The topic is cell cycle and signaling, foundation of life and links to cancer. Today we are going to talk about two fascinating cellular processes, the cell cycle and the cell signaling. And both of them are very essential for life as they govern how the cell grows, divides and communicate. However, these processes, when gone wrong, can lead to disease like cancer. And by the end of this presentation and discussion, we'll understand how the system works and what are the importance of this system. Feel free to ask any questions after the presentation. So the cell cycle is distributed in three phases, G1 as G2. The G1 phase is responsible for the growth where the cell gets bigger and prepares the DNA for replication. TS, also known as synthesis phase, copies the amount of DNA so that each daughter cell can complete the set. The third is G2 phase where the cell prepares for division and then checks for any error. Like any good factory, the cell cycle has also checkpoints to ensure that everything is running smooth and in quality. The first would be the G1 checkpoint, which is the decision point. The purpose for this checkpoint is to know the restriction point, determine whether the cell is ready to proceed with the DNA replication or not. This checkpoint occurs late in the G1 phase. It checks the nutrients available, the cell size, the growth factors, which are external signals, example hormones or mitogens present to promote the progression, and the DNA integrity, is the DNA damage free or not. And the mechanism for this is, if the conditions are favorable, cyclin D binds to CDK4 or 6 phosphorylating the RB, also known as retinoblastoma protein, and release E2F transcription factor, and this drives to the transcription of genes necessary for replication. If the DNA is damaged or it is detected so, the P53 is activated, inducing the expression of P21, a CDK inhibitor. This prevents the RB phosphorylation and holds the cell cycle. And the outcome for this is if it passes, the cell enters S phase and becomes the DNA replication, or else if it fails, the cell is paused, allowing the time for DNA repair. If damage is irreplaceable, that cell may go apoptosis. Now let's move forward to G2 checkpoint, also known as the quality control after the DNA replication. The purpose of the G2 checkpoint is to ensure that the DNA replication in the S phase was accurate and that the cell is ready to enter the mitosis. So, the DNA replication completeness, has the entire genome been copied or not? The integrity of the DNA, is there any error or in the replication or not? And the cell size is large enough to divide or not? The mechanism is that it relies on the cyclin B CDK1 complex, also known as maturing promoting factor MPF. If the DNA is detected or sensors like ATM or ATR kinases activate the CHK1, CHK2, which inhibits the activation of CDK1, preventing entry into mitosis. Proteins like BRCA1 and P53 may also be involved in repairing the DNA and hold the cycle. Let's understand the cell signaling, which is how the cells communicate. Cells constantly receive signals from their environment and other cells. These signals guide their actions like when to divide, repair damage, or respond to threats. Imagine you are in a relay race. The cell's signaling works like passing a baton from one molecule to another, eventually leading to an action or response. Cell signaling has three main steps. The cell signal reception, a signal molecule like a hormone binds to a receptor on the cell surface like a key fitting into a lock. Cell transduction or signal transduction, in which inside the cell the signal is converted into a series of steps, much like how the relay race works. And the response, the cell performs an action like dividing, making a protein, or stopping the growth. For example, when you eat, your pancreas releases insulin, which signals your cells to take in sugar from the blood. There are various pathways, but over here we are going to talk about two of them. The first one is the first pathway is G coupled receptor pathway. Example over here we are going to take about adrenaline or epinephrine signaling. So the reception steps over here are adrenaline binds to a GPCR on the surface of the target cell. The receptor undergoes a conformational change and activates a G protein by causing GDP to be replaced with GTP, which is the active version. The activated G protein activates an enzyme called adenylyl cyclase. The adenylyl cyclase converts APT into second messenger called CAMP. The CAMP activates protein kinase A, which phosphorylates various targets to amplify the signal. In response, the liver cells activates enzymes that break down the glycogen into glucose, increasing the blood sugar levels. And in the heart cells, the pKa increases the heart rate and the contraction strength. So the termination step for this is the signal is turned off by hydrolysis of GTP on the G protein, degradation of CAMP by phosphodiesterase, and deactivation of GPCR. The another pathway which I'm going to talk about is MAP kinase or mitogen activated protein kinase pathway. The example over here we are going to talk about the growth factor signaling or the EGF. The epidermal growth factor binds to the RTK, causing receptor dimerization and photophosphorylation of tyrosine residues. And the transduction would be adaptive proteins like GRB2 and SOS are recruited to the receptor and activate the RAS, a small GTPase. RAS or RAS activates the MAP kinase cascade. RAF, MAPK all amplifies the signal and with ERK phosphorylating multiple target proteins. The response to this is in the gene expression, the ERK phosphorylates transcription factors like ELK1, promoting genes involved in cell division and proliferation. Cell cycle progression, the S phase. How can we terminate this? So the hydrolysis of GTP on RAS again dephosphorylates MAPKs by phosphatase. And so over here we are going to also talk about P53, also known as the guardian of genome. The P53 is a transcription factor encoded by TP53 gene. It monitors cellular stress, particularly DNA damage, and acts as a key decision maker for cell's fate. The role for this in the cell cycle is the DNA damage response. Where the DNA damage occurs, the P53 is activated by upstream kinases like ATM and ATR. Activated P53 induces the expression of P21, a cyclin dependent kinase, which is an inhibitor, and that holds the cell at the G1 or S checkpoint to allow the time for DNA repair. If the damage is too much or it is irrepressible, it goes to apoptosis as the P53 activates pro-apoptotic gene like BAX and PUMA, initiating programmed cell death to prevent the progression of damaged cells. And this leads to senescence as a permanent state of growth arrest or to prevent potential cancerous cells from dividing. In cancer, the P53 is mutated and inactivated with over 50% of cancers allowing cells with damaged DNA to divide unchecked. Loss of P53 removes critical barrier to tumorigenesis. And the second protein over here we are going to talk about the retinoblastoma, also known as the RB, and it is the gatekeeper for the G1 and S transition. The RB protein encoded by RB1 gene is a key regulator of the cell cycle and the tumor suppressor. The RB binds and inhibits E to F transcription factors, which are necessary for the expression of gene required for DNA replication. In its hypophosphorylated state, also known as the active state, the RB prevents cell from entering the S phase, acting as a checkpoint control. If it is released in response to the growth signals, the CDKs or the phosphorylated RB inactivates. The phosphorylated RB release E2F, allowing the transcription of gene required for DNA synthesis and cell cycle progression, and preventing the uncontrolled division. In cancer, loss or inactivation of RB results in uncontrolled activity of E2F, leading an unchecked progression through G1S checkpoint. Mutations in the RB1 gene are associated with the cancers like retinoblastoma or an eye cancer in children, osteosarcoma, and other malignancies. And so the relationship between P53 and RB in cancer prevention goes like this. The P53 and RB act in parallel pathways to suppress tumor development. P53 monitors DNA integrity and holds the division if damage is detected, while the RB ensures the cell division only occurs in response to appropriate gene signaling. And together they maintain a tight control over cell proliferation. The oncogenic disruption. So now we also know that what if the things goes wrong when P53 and retinoblastoma are not acting properly, correct? In many cancers, both P53 and RB pathways are inactivated either by mutation or by action of viral oncogene, for example HPV or human papilloma virus, which produces a protein that degrades P53 and inactivates RB respectively, promoting the cervical cancer. So cancer is a prime example of what happens when this system fails, and cancer cells bypass checkpoints and often create their own path signals leading to tumors. And so the examples over here, we would like to talk about the breast cancer, in which breast cancer cells have too many HER2 receptors, making them hypersensitive to growth signals, and this leads to uncontrollable cell division. The skin cancer goes something like this. When the UV radiation damages the DNA in skin cells, it damages isn't caught at a checkpoint, these cells can become cancerous. Understanding the cell cycle and cell signaling has led to major breakthrough in medicines. The checkpoint inhibitors, where these drugs are these are the drugs that help the immune system recognize and attack the cancer cells. Gene editing tools like CRISPR, these can correct genetic errors in cells, potentially curing the disease at their root. And these advances give us hope for treating not just cancer, but many other diseases caused by faulty cellular processes. In summary, or to sum up, the cell cycle and cell signaling are vital for life, ensuring cell growth, divide, and communicate properly. When these systems fail, disease like cancer can occur. However, our understanding of these processes is leading to groundbreaking treatment that could change the future of medicine. Thank you for listening, and I am happy to take any question.